So Kelly received her BA in neuroscience from Boston University back in 2014. Then she worked with David Sommer and Swati Kiran, and she developed great interest in neuroimaging and neuroplasticity. She did her PhD work at Georgetown University with Peter Harteltaub and Lisa Newport. And her focus was in the language system and plasticity in the language system in healthy development and after stroke. So this is what she will be talking about today. And she's currently a postdoctoral fellow at Georgetown University. She graduated recently. It was last summer.
Last June.
Okay. So welcome, and the floor is yours. Okay.
Thank you so much, Maria, for introducing me, and thank you, Swati, and everyone for inviting me to give this talk today. Is the volume okay? Can you guys hear me? Okay. So I'm very excited to tell you a bit about my thesis work that I did at Georgetown, as Maria said, with my co-mentors, Alyssa Newport and Peter Turkeltaub, and also Anna Greenwald. This is a picture from my defense last year, which was like the best day of my life. So happy to talk about that with any students who are interested. So today, as Maria mentioned, I'm going to talk about this thesis work, which was on language system plasticity in healthy development and after perinatal stroke. For the last year, I've been returned to adult stroke and have been looking into plasticity in adults again, and I'd be happy to talk to anyone who's interested about that. Oh, okay. So this is just an overview of the talk today. So the big picture question that many of us are familiar with is that lateralization is an important feature of language system organization, and it's one that actually appears to strengthen throughout the course of development. And so for my dissertation work, we focused on this question beneath this, which is, what are the consequences on the brain organization of language processing when left hemisphere regions are unavailable from birth? And so we pursued 3 studies that I'll talk about. I'll step through these today. The first one asked, to what extent does language engage homotopic right hemisphere regions in the typical brain, even in its mature state? So homotopic meaning mirror image or parallel regions. We then went on to study 2 different investigations into perinatal stroke, which I'll introduce a little later in the talk. So the second study examined, is language engaging homotopic right hemisphere regions after a left hemisphere perinatal stroke? So that's around the time of birth. And then finally, does language processing overlap with the typical functions of homotopic right hemisphere regions after a left hemisphere perinatal stroke? And so just to give a little bit of background here, I know everyone is pretty familiar with this already, but I think it's nice to kind of revisit the evidence for why we know that this is true. So language is left lateralized in most adults. From stroke and aphasia, we know that when a stroke impacts the left middle cerebral artery, which I'm showing in yellow in this picture at the top, the consequence almost invariably is a chronic language impairment or aphasia. However, that outcome is pretty rare if that impacts the right middle cerebral artery. And so this is some early evidence that indicates that the left hemisphere seems to be critical for language processing. And with more modern neuroimaging tools, we've been able to actually map where specific problems or impairments in language processing map onto different lesion locations. So I'm showing one example of that beneath this. There's also interesting evidence from patients who undergo split brain surgery or corpus callosum transection. And so when some people have intractable epilepsy, they can undergo a procedure where their corpus callosum is divided. And so a common paradigm is to show a word to one side of the visual field. Since the optic radiations are still intact, they can bring that information to the contralateral hemisphere. And then you can ask questions about what each hemisphere can do on its own without transcallosal communication. And so this is just an example of one typical paradigm. So the general finding is that when you show a word to the left visual field, the right hemisphere is often incapable of producing a verbal response. It can do something like gesture, or you can draw a picture, for example, here with face. Whereas the left hemisphere, when you show a word to the right side of space, appears to be okay in terms of its normal verbal function. And so this is pretty compelling evidence that the left hemisphere is critical for language processing and the right hemisphere is pretty limited without the ability to interact with the left. The WADA test has also provided some interesting evidence. So again, in patients with epilepsy who are about to undergo surgery to remove brain tissue, in the WADA test, you can anesthetize each hemisphere one at a time and just probe what verbal abilities the patient still has when one hemisphere is anesthetized. And so this is just one example of some data that were collected. And in this sample of 229 patients, 80% had left hemisphere lateralization for language. And then finally, and of interest to a lot of us especially, fMRI has provided really important tools to be able to examine language organization in healthy adults, and in particular, which regions seem to be active for language processing. So not just whole hemisphere, but particular regions. And so there are a lot of different language paradigms out there. One is forward over reverse speech, which is going to be the premise of a lot of what I discussed today. There's also mental verb generation, so many different types of language tasks. And the very common result is robust activation in the left hemisphere and not very much going on in the right, certainly at the group level. And I'm also just showing at the bottom here a contrast of in congenitally deaf individuals, sign language compared to layered backward sign to show that it is in fact still left lateralized in most of these individuals. So it really is something about language processing and not necessarily anything specific to auditory speech processing that appears to engage the left hemisphere. It's actually pretty weird that language is lateralized, and it's even weirder that it's so commonly left lateralized. So symmetry in general is advantageous. Asymmetry is usually the exception. As mammals, we move linearly through our environments. We have symmetrical limbs controlled by bilateral somatomotor cortices. And then bilateral sensory systems to detect and react to incoming information from every angle. The language system depends on sensory and motor systems for input and output. So you would think that language processing should be somewhat bilateral. But it's important to remember as well that the brain develops along 4 axes rather than 3. So we have rostrocaudal, dorsal ventral, and then each hemisphere actually develops along its own medial lateral axis. So there's perhaps a little more room for asymmetry in this direction. Nonetheless, we still develop pretty symmetrical brain structures. And I've just included a caveat here that, of course, language processing is more than just one thing. There are many sub-processes, and the third part of the talk gets into that quite a bit more. And so there's some debate over exactly which processes are more lateralized than others, but in general, the processes I'm going to show you today quite like left lateralized. And so now that I've spoken quite a bit about how language processing is left lateralized and it's unusual, I'm actually going to tell you that it appears to be more bilateral in young children. So there was some recent work that was done by Peter, Alyssa, Anna, and their colleagues that looked at children of different ages, so 4 to 13 years old, as well as adults 18 and older. And they performed an auditory sentence comprehension task in the scanner. And you can see here that in these group average activation maps from youngest, middle, oldest to adults, the left hemisphere activation is pretty stable. It's in the same places, it's pretty strong, but the right hemisphere doesn't show a lot going on in any of these age groups. However, when they took a closer look at significant activations within individual participants, so not group averages, but where there were significant activations in individuals within each group, they found that there were a greater number of significant activations in the right hemisphere of the youngest children. The youngest are 4 to 6-year-old. So that's quantified in this bar graph here. So you can see that there are a greater number of significant activations in the right frontal cortex and right temporal cortex in the youngest age group compared to the others. And relatedly, they actually found a significant decline in the amount of activation related to age in the right frontal cortex. And so it appears that there's something happening, at least in the right hemisphere with age, where perhaps it's more strongly active during language processing when you're very young, and then it transitions to being less involved as you get older. So the first study in my dissertation focused on this question: to what extent does language engage homotopic right hemisphere regions in the typical brain, even in its mature state? And so we published this paper last year. I'm going to talk through some of the main results from it now. And if anyone's going to the SNL conference in October, I'm going to give a little talk on it again. This was a really fun study to do, and I'm happy to answer questions about it. It was kind of like a side quest that we ended up doing, and it proved to be really important for the rest of my work. So what does seem to be changing in the right hemisphere of adults? There was some early fMRI work done by Justin Carpenter, which I'm showing in the bar graphs to the left, where they had adults in the MRI scanner reading sentences that varied in complexity. And what they found in Broca and Wernicke's areas were a staircase sort of increase in activation based on how complex the sentences were. And when they looked in the homotopic areas in the right hemisphere, they found that the activation was much lower. So there was a lower mean number of voxels activated, but there was still a staircase pattern that seemed to reflect an increase in response to the increase in complexity of the sentences. And so they ended up calling this pattern a weak echo, perhaps of what's going on in the left. And this is really interesting, but not much work has been done to follow up on what the right hemisphere is doing in this regard since. Most of the reason for that is because in fMRI work, we're often focused on the brain regions that appear most strongly active or most consistently active or both. And there's good reason to do that. But it is also not an unusual finding to see that sometimes people report that they found a subthreshold response in the right hemisphere that seemed to be similar to what they had found in the left. But it's rarely the focus of the results. And so I'm just showing an example of that from Quillen et al. 2021, where they had a language task that involved asking, does the pair of words go together? And then a perceptual control task, which asked, is the pair of symbol strings identical? So just visually comparing them. And you can see here the same type of echo situation. So in the left posterior superior temporal sulcus and in the right, you see that the left is more active than the right, but that there's a similar pattern to both the language task and the control task. And then again, in the left and right inferior frontal gyrus, we see that the right is much weaker, but that the pattern is very similar. And so in this study, we wanted to know, are homotopic regions in the right hemisphere still weakly active in adults? So on one hand, the activation may become weaker with age, but also disperse. So it's no longer sort of intact regions that are there, but weakly active. They may also, over time, have become more involved in other cognitive functions, less dominantly involved in language. Perhaps dominantly is not the right word, just less involved in language. Alternatively, the activation may become weaker, but still be intact. And so that we would think is something like a weak shadow of perhaps the stronger activation that had been there earlier in life. I've had a lot of interesting conversations about the term weak shadow, and I'm definitely happy to hear other people's interpretations of that. In general, I really like when these kinds of presentations are interactive, so please interrupt and ask me questions. I'm going to get through the introduction in a little bit, and I'll definitely pause after each study. So what do I mean by strength of activation? So here I just have sort of a toy example where we have arbitrary units of activation on the y-axis, and there's 2 participants, and we have 7 different brain locations that are active in each person. And so we see here that the areas of greatest activation for each person are the same. It's areas 1, 3, and 5. However, participant 2 is a much weaker activator. And so when we use a conventional statistical threshold, say p less than 0.001, what we would find is just the regions that beat that threshold for everyone. And so for participant 1, we would conclude that the regions active are 1, 3, and 5, whereas participant 2, we would only identify location 5. And so that might be what you want to do, depending on your research questions. You can also imagine this being the left and right hemisphere. So you have regions that are similarly active in both, perhaps, but you can't actually identify which regions are similarly active if all that you care about is just the strongest activity. And so if your question revolves around the spatial location of activity without requiring the activity to be very strong, you can take a different approach and say, just give me the top 3,000 most active voxels for each participant. And then you can see here that we would conclude the active locations in participant 1 are 1, 3, and 5. And in participant 2, their most active locations are also 1, 3, and 5. And so again, we can think about this being the left and right hemisphere. So this is a way of comparing the spatial location of activity that is strong or weak. And so once you have all of the voxels that you want to consider, the activation maps, say left and right hemisphere, or map 1 and map 2 here. How you actually go about computing the spatial overlap is a second question. And so one way of doing that is the dice coefficient. And so here we might have 3,000 voxels in map 1 and 3,000 in map 2. We can flip map 2 onto map 1 and then take a look at how big that intersecting area is. And so the dice coefficient calculates this. So it looks at the size of the overlapping area as it relates to the size of the 2 initial maps. And so because we cared about making sure we're looking at the same amount of activation, so again, around 3,000 voxels or so in the left and right, the X and Y in the denominator here are the same size.
Can I ask a question before you proceed?
Please.
So this is for each person, you have 2 maps, one on the left and one on the right hemisphere.
Mhm.
And you do this for every subject.
Yeah. So I'm going to step through that now, which is a great segue. So I'm going to step through this pipeline in some amount of detail. I never quite get it right, so feel free to ask follow-up questions. And this is the general pipeline for the following 2 studies as well. So I'm spending a good amount of time on the top voxel approach because it's important to understand the rest. So for this particular study, as Swati just mentioned, what we did was we took each participant and we flipped their right hemisphere into the left hemisphere space. So we flipped their right hemisphere and we normalized it to left hemisphere MNI anatomy. That put them both in the same space to be able to overlay them and compare. We wanted to, so this is for forward over reverse speech. So again, this task was an auditory sentence processing task. We looked within regions of interest, which for this study were functional regions based on the neurosynth meta-analysis language keyword, so studies that have reported the word language in their results. And then the way that we go about calculating the top voxel levels, we end up with 4 cutoffs, and that's just a rigor note, so it's just to make sure we're not picking one arbitrary number of voxels. So we take the unthresholded T-map for each participant in their left hemisphere within each of these ROIs. And then we threshold at 4 different levels. So we'll say for the most lenient, it's P less than 0.01. So we just say how many voxels are active at P less than 0.01 for all participants. And we just take that average, and that average at P less than 0.01 in the frontal lobe was around 3,000, and in the temporal lobe, it was 2,588. And then we do that at 3 other P thresholds. So that gives us our 4 top voxel cutoffs in the frontal lobe and in the temporal lobe. So we then have 2 maps of equal size for the left hemisphere and the right hemisphere for each participant at each of those top voxel cutoffs. And so we can then take these maps of equal size, overlay them, and calculate the dice coefficient to ask how overlapping those maps are. I'm going to pause there. Was that pretty clear? We can also revisit this. Okay, so what we found when we did this was that we looked in each age group. So 4 to 6-year-olds are in the salmon color, 7 to 9-year-olds are in green, the blue is the 10 to 13-year-olds, and adults are in purple, and we have the frontal ROI and the temporal ROI. So the y-axis here is the dice coefficient mean across levels. So we calculated the dice coefficient at each of those 4 levels per person, and then we just averaged across because there was no difference across the levels. And so what we found here is that there's no statistical difference among any groups. So adults were not, in fact, significantly lower in terms of how homotopic their own activation was compared to any of the younger child groups. The white dashed line here shows what random chance would be in terms of random arrangements of the same number of active voxels in the same size space. So taking the whole ROI, arbitrarily assigning 3,000 or so voxels as being active, and then shuffling those 10,000 times, how much would those randomly shuffled voxels overlap? And so all of our age groups beat that random chance. We also did a slightly more rigorous comparison, which I'm not showing here, but is in the paper. And so the dice values for their own left hemispheres with their own right hemispheres were greater than the well-matched reference comparison of their own right hemispheres with everyone else's left hemispheres. So rather than having randomly shuffled voxels, we actually used real activation maps. And so I didn't know exactly how that was going to pan out, since it's going to be pretty similar. You'd think if they're quite homotopic in the left and right, they may also be very consistent in terms of each other's lefts and rights. But in fact, their homotopicity was even greater than that. I think we invented the word homotopicity, but I might accidentally say it a lot of times. So, okay. And then these are penetrance maps, which are a little different from activation maps. This shows the consistency of where participants were active. And so the areas that are darker blue have a greater percentage of participants that had activation there, or top voxels there. And so you can see here that for each of the age groups, so we have the youngest to the oldest, the activation consistency is in similar locations. And so the kind of wrap up here is that in infancy and in first years of life, language is more bilateral in young children. And then something changes in right hemisphere regions in which they become slightly less responsive during language tasks. And then by adulthood, language appears to be left lateralized in the majority of adults. So in summary, based on the Oluladay work, what appears to change is that activation weakens in homotopic right hemisphere regions. Based on the current work, what does not appear to change is that these regions still have a spatially intact weak response to language processing. And so in conclusion, it seems that right hemisphere regions that once supported language processing early in life may still be available to some degree. That being said, they don't appear to be fully supporting language after a left hemisphere stroke in adulthood. And if these regions cannot perform language processing, then why is the space still being used this way? So these are just kind of questions that remain. I'd be happy to hear your thoughts about those. That's the end of the first study. So next I would move on to the perinatal stroke work. Does anyone have any questions right now? Okay, I'll move on, and I think it should all kind of work together. So maybe people will have questions at the end about all of it. So in the next study we examined, is language engaging homotopic right hemisphere regions after left hemisphere perinatal stroke? So perinatal stroke is a cerebrovascular incident near the time of birth. The prevalence is about 1 in 3,500 term births, and most of them, about 80%, are arterial ischemic. They're most commonly impacting the middle cerebral artery, which is again in yellow here, very similar distribution to what we see in adult strokes that cause aphasia. 2/3 of these strokes, for some reason, impact the left hemisphere, and that's still kind of unknown in terms of cause. The cognitive abilities in these individuals are in the normal to low normal range when tested as adolescents and adults. So this has been shown in several behavioral studies, but what I'm showing you here is also some data from our lab's work, which you can also read a bit more about in the PNAS paper that Lissa just published earlier this year, last year, I guess now. So here is just a graph showing the IQ measure that we use. So there's verbal IQ in blue, performance IQ in red, and full scale IQ in purple. And so in the pediatric stroke research study, which is done at Georgetown that Lissa leads, we have left hemisphere stroke participants, right hemisphere stroke participants, and the control participants are actually the healthy siblings of the stroke individuals. And so you see their healthy siblings are very high performing. The solid black line is the normative mean. And the stroke groups are a little lower than that, but they're still within 1 standard deviation of the normative mean. And so considering the amount of tissue loss this early in life, it's really unbelievable how well they perform. And in this study, we collect a lot of different language tasks that really try to carefully characterize the abilities of these kids. And so here I'm showing you the TROG2 comprehension of syntax. So they'll hear a sentence like, the duck the ball is on is yellow, and they'll be asked to point to a picture. And so it just probes sentences of varying complexity. And you can see here that compared to the healthy sibling controls, both of the stroke groups perform extremely well on these tasks. And again, there's more detail about the behavioral measures and the cognitive and language abilities in the PNAS paper. And so especially important for the rest of what I'm going to show you today, language does appear to be developing within the normal range, low to normal range in these individuals. And when it does, it seems to be organizing in right hemisphere regions. And so here are a few examples at the top. I have 2 images of individuals from our stroke cohort that have different strokes in different locations. And you can see that the activity is in right hemisphere regions that appear to be homotopic to where you would expect it in the left. And then there's also 2 group maps from a study by Guzetta and Telema that show right lateralized activation for language processing after left hemisphere perinatal stroke. And so unfortunately, group average maps do not reveal where individual participants, whether you have a stroke or otherwise, may recruit regions in an idiosyncratic fashion. So they tend to show areas that are strongly active and also consistently active across individuals within a group. So we don't necessarily see where individual participants may be recruiting additional areas. And that is especially interesting if you expect that someone might have an alternative brain organization after something like a stroke at birth. And so in the following study, we examined, 1 is the activity in individual stroke participants, left hemisphere perinatal stroke participants, right hemisphere regions, a mirror image of typical left hemisphere activity. So it appears that based on the group images that they are, but is it true that in individuals, there's precise organization in the right hemisphere in mirror image regions? And then second, especially because of the results from the first study, we also wanted to know whether language activation in the right hemisphere of the perinatal stroke participants aligns with areas of weak activity in the typical right hemisphere, or if these regions are more similar to a typical left hemisphere. And so you might imagine that in order to develop a language system in the right hemisphere, it may need to be more similar to the typical left hemisphere. And even though we just showed that the left and right hemisphere are quite homotopic across age, there's still a margin in which they're not exactly the same. They're not perfectly overlapping. And so perhaps that margin is actually quite important for organizing a language network. Maybe there's something there. And so this is the overview of the analysis pipeline again, and I'm just going to point out what's different within this study. And so here we have the stroke participants as well as controls, and we're going to be analyzing the right hemispheres of the stroke participants flipped into the left. And so this allows us to line up their activation in the right hemisphere with the left hemispheres of controls. And then we're also going to compare their right hemispheres to the right hemispheres of controls. We're going to be using anatomical regions of interest this time, so the whole frontal lobe and the whole temporal lobe, and that allows us to sort of cast a wide enough net to see if there's any areas of activation that fall outside of very specific canonical language regions. For the top voxel levels, we averaged across all participants in their dominant hemisphere, so the right for the stroke patients and the left for controls. And we had those 4 cutoffs again. So we were ultimately comparing maps of equal size for the control left hemisphere, the control right hemisphere, and then the stroke participants' right hemispheres. And so when we performed this analysis, what we found was that the language processing, the activation in the right hemisphere, was consistently in homotopic regions, so regions that were pretty precisely parallel to the typical left hemisphere regions. And so I'm just showing you all the way on the right snapshots of the different stroke sizes and locations in our group, so you can see that they're quite variable. Interestingly, in contrast to what we see in adult stroke, stroke size does not often correlate with behavioral outcomes or whether language organizes in the right hemisphere. That point's a little more complicated and has a lot to do with how early you had your stroke. However, yeah, so it's interesting that stroke size does not appear to be as big of a factor as it is in adult stroke. Seizures are something that do tend to relate more than the stroke size or stroke side in terms of language organization. But anyway, I digress. So what I'm showing you on the left here in blue is left hemisphere activation for control participants compared with all other control participant left hemispheres. So this is control lefts compared with control lefts, and that is kind of an upper bound for how similar we think individuals should sort of be for our task activation. In purple, I'm showing the right hemisphere activation for our left hemisphere perinatal stroke participants flipped into the left hemisphere and compared with each of the controls. So this is really our comparison of interest. This is how similar their right hemisphere activation is to the lefts of controls. And then finally, just because it was interesting, we also examined the right hemisphere activation for left hemisphere perinatal stroke participants compared to the other perinatal stroke participants to see if they were just as similar to each other as maybe they were to the healthy controls. And so what we found is that when we flip the right hemisphere activation for stroke participants into the left hemisphere and compare it to controls, the overlap was high enough to be in the same range as the overlap of controls with one another. And so in the middle, I'm showing you z-scores, which I think drive this point home very clearly. So a score close to 0 means you're getting like very, very close to the control mean with one another. And so the closer you are to 0, the more similar you are to the distribution of similarity among control left hemispheres. And so our stroke participants were, for the most part, all within 2 standard deviations of that control mean. And so this was really interesting. We then examined whether sentence processing areas were more aligned with the typical left hemisphere than the typical right hemisphere areas. And so if the activity in the right hemisphere of stroke participants is well aligned with the weak areas of activity in the right hemisphere of controls, then perhaps the right hemisphere recruitment is not necessarily a change in the developmental functional layout. So if they're just kind of recruiting the areas that are weakly active in adults and maybe more active during typical development in childhood, maybe it's not a change that's due to post-stroke plasticity. Maybe they're just leveraging these existing weaker in typical development regions in the right hemisphere. And so the light blue bar and the purple bar, the light purple bar, are the same from the previous slide. So the light blue is the left hemisphere activation for control participants compared with all other control left hemispheres. And the lighter purple is the right hemisphere of the stroke participants flipped into the left and compared with all the control left hemispheres. And so the 2 added comparisons here, one is just a reference point of what does the left hemisphere activation for control participants look like compared to flipped right hemispheres for other controls, just to say, is it something about the flipping process? And you see that that dark blue bar is much lower than the purple bar.
Can I just clarify, that's the light blue bar is the gold standard, that's left hemisphere
Yes.
activation in controls.
That's right. That's right. And so the light purple bar is the stroke patient's right hemispheres compared to left hemispheres in controls. So that's similarity to a typical left hemisphere. The dark purple bar is really the one that's of interest that's added here, which is the right hemisphere of stroke patients compared to the right hemispheres of controls. And so the light purple compared to the dark purple, we see that they are much more similar. The right hemisphere activation in the stroke patients is more similar to the typical left hemisphere than it is to the typical right hemisphere. Oops. And then you can see in these penetrance maps at the top, this is just showing the overlap of the top voxels in the stroke participants' right hemispheres overlapped with the control participants' left hemispheres and the control participants' right hemispheres. And so areas that are darker have a greater number of stroke participants with overlapping activation there. And you can see that there's more consistency in the overlap with the typical left hemisphere compared to the typical right hemisphere.
So can I ask one more question?
Mhm.
The darkest point is in the right hemisphere frontal, but that is really showing the overlap with the weak right hemisphere of the controls, right? That dark left.
The dark left, so that is where stroke participants were very consistently overlapping with control participants.
In the control participants' right hemisphere. I'm sorry, that's left hemisphere.
Control left, yeah.
Control left is the gold standard.
Well, this is the stroke patients compared to controls.
Yeah.
In these brains here. Yeah.
So the contrast is always controls here.
Yes, yeah. And here I'm showing for each of the hemispheres and groups individually. Sorry, the zoom bar is blocking it. The one on the left is the control left hemispheres with one another, so controls with controls. The one in the middle is the control flipped right hemisphere. So again, controls with controls, but it's the right hemispheres shown on the left. And then the one on the right is the stroke participants flipped right hemispheres. So they're right hemispheres with one another. And here I'm showing the areas that are in brighter white have higher consistency. These are those same maps thresholded for any regions that have more than 27% of the participants with overlap there. And I did that so that I can then compare them to one another. And so I just made this figure recently because a reviewer asked for it, and I actually think it helps clarify things a lot, but I would be welcome to hear whether you disagree. And so the major question here that we were trying to address is really about the central figure on the bottom and the one to the right. So control left hemisphere with the stroke participant right hemisphere, and control right hemisphere with the stroke participant right hemisphere. And areas where they have in common would be in pink. And so you see that there's a lot of pink with the control left and the stroke participant right, whereas you don't see as much with the control right and the stroke participant right. And so the areas that are purple in the image to the right, where it's control right hemisphere compared to the stroke right hemisphere, are the areas where the stroke right hemisphere had more language activation, or more consistency among the participants for their language activation. And that's sort of the swath of frontal cortex, for example, where you see a lot of pink when you look in the map comparing to the left hemisphere. And so this is just generally trying to get at the point that there are many regions that are shared between the right hemisphere after a perinatal stroke and the left hemisphere and the typical brain. And the regions are not as consistently shared between the right hemisphere of the typical brain and the right hemisphere after perinatal stroke. So from this work, we conclude that sentence processing consistently recruits right hemisphere regions that are homotopic to typical left hemisphere language regions in individual stroke participants. And that sentence processing in stroke participants may be recruiting a pattern of areas in perisylvian cortex that is more similar to a typical left rather than the typical weakly active right hemisphere areas. This particular pattern of recruitment might be important in a dominant hemisphere for language processing. And it may also leverage early life plasticity privileges that might be unavailable after a stroke in adulthood, just because they do appear to be organizing something like a typical left hemisphere in the right and not perhaps existing weakly active areas like we have seen in the adults. So next, I would move on to the last study, unless anybody has questions so far. Okay, that's fine. Okay. So the last study that we looked at examined whether when language processing activates right hemisphere regions after a left hemisphere perinatal stroke, does it overlap with the typical functions of the homotopic right hemisphere? And so, as we mentioned at the beginning, language contains several sub-processes. I'm just showing you two visuals of that here, and we're not going to get too in the weeds with it, but generally speaking, one way to delineate these processes that people discuss are segmental processes versus suprasegmental processes, where segmental are like the small bits of information that are quickly modulated, things like phonetics, phonemes, morphemes, whereas suprasegmental is things like prosody or changes in pitch and harmony of someone's vocal tone. So for example, if you can tell from my tone of voice, whether I'm mad or happy or a little nervous, that would be the prosody, which is above and beyond what the actual content of the words that I'm saying to you. And so those types of suprasegmental processes tend to be right lateralized. And so there's some interesting work that shows that sentence processing and emotional prosody exhibit opposite lateralization in the healthy adult brain. And so this is some imaging work that was done by Anna Greenwald, where healthy adults completed a sentence processing and emotional prosody task in the scanner. And so they heard sentences, and they also had a control condition of reverse sentences. They also heard sentences that were spoken in a happy, sad, or angry tone, and then reversed versions of these emotional sentences. And so at the top, you see that the neutral sentences minus the reverse neutral sentences show the left lateralized pattern that we've shown a few times before. When she compared the emotional to neutral conditions, it was right lateralized. These are group maps. So right lateralized activation, and it was even more right lateralized when she subtracted out the acoustic control conditions from each. So you see in the bottom figure here, in particular, that very strong temporal activation that is bilateral seems to be eliminated when you have a good acoustic control. And for anyone that's used to looking at lateralization indices, you can see for individual participants, the blue dots for the Sentence processing over reverse sentence task are very left lateralized. And in red here, we have the emotional minus neutral with the controls in red. And so those are very right lateralized. There's also a lot of evidence in adult stroke that right hemisphere strokes cause something called aprosodia rather than aphasia. And so aprosodia is a prosody processing impairment. These two studies that I'm showing you here involve different types of speech stimuli. So there were words, monosyllables, and also asyllabic stimuli. And what I just want to show you is that except for the word condition in the left on the comprehension task, the right hemisphere stroke group, which is the bottom line here, performs worse than the left hemisphere stroke group and the control group. And so this isn't a comprehension task where they would listen to a speaker's voice and then select which line drawing of a face and label was the best match for the emotion they'd heard. And then even more markedly in the discrimination task where they heard pairs of stimuli and judged whether they heard the same or a different emotion, the right hemisphere stroke group performed much worse than the left hemisphere stroke group and controls. And so these are adults. But this leads us to the question, can these processes recruit the same neural territory under extreme circumstances, for example, after a left hemisphere stroke at birth when language moves to the right hemisphere, but you're still developing? Or will they still be driven to segregate within right hemisphere perisylvian cortex? So because we have these systems in parallel hemispheres in the typical brain, if you have to fit everything into one hemisphere, do they end up forming subregions? And so for this study, we had the auditory description decision task, which is the same task that had been used in the prior work. So they heard sentences that either made sense or didn't, like a big gray animal is an elephant, or something you sit on is a triangle, and they would press a button if the sentence made sense. And then they also heard these sentences played in reverse as a control condition, and sometimes there was a tone at the end, and they'd press a button when there was a tone. We also had the emotional prosody decision task, which is similar to the one that Anna used in her work with the healthy young adults. And so there were emotional speech blocks where they would hear a sentence like, "Mom poured me a huge glass of milk," said in an angry, happy, or sad tone. And then they saw a visual cue at the end that was either a boxing glove, raindrops, or a sun, and they'd press a button if the visual cue was congruent with the emotion that they'd just heard. The control task for this was neutral speech blocks, where they heard similar sentences, but they were told to pay attention to what was happening in the sentence. So someone is going somewhere, receiving a gift, or eating something, and they saw a visual cue that was a car, a gift, or a fork and plate. And they pressed a button when the visual cue was congruent with what was happening in the sentence. And so for emotional speech here, we're subtracting out activation during neutral speech blocks. So to answer this question, can sentence processing and emotional prosody recruit the same neural territory in the left hemisphere, perinatal stroke, right hemisphere? We have two potential outcomes that we wanted to dissociate between. So the first was perhaps sentence processing can be mirrored into the right hemisphere and recruit overlapping territory with emotional prosody. Perhaps that's fine. You don't actually need separate territories to process these things. Alternatively, we may observe functional partitions in these right hemisphere regions in order for both processes to be supported and for them to develop successfully. And so just to visualize what that might look like, this isn't real data, but we have at the top for a control subject, we may see in blue, the flipped sentence processing activation into their right hemisphere, overlaid onto their emotional prosody activation in the right hemisphere in red. And when we look at the overlap, we would see in magenta that there's quite a lot of overlap in the frontal and temporal lobes. And then in stroke participants, we might see that both tasks recruit the same set of regions generally, but they sort of find ways of shifting around each other so that both can be supported in the same general set of regions, but that the overlap is actually quite minimal. So this would be something along the lines of subregions, but just separate territories in some capacity. And so this is what we sought to test. But first, we wanted to look at the extent of activation for each of these tasks. So if sentence processing recruits a left hemisphere-like set of regions in the right hemisphere after left hemisphere perinatal stroke, which was our finding from the prior study, does it recruit right hemisphere perisylvian regions more dominantly or to a greater extent than emotional prosody? So perhaps the way these things fit together in the right hemisphere is that language just activates everything and sort of takes over. In which case we might see a lot more activation for that task compared to control participants in just the one hemisphere of the stroke participants. Or we might see that greater activation for the speech processing always relates to less activation for the prosody processing within individual stroke patients. So we just looked at both of those things. When we looked at the number of active voxels in the frontal and temporal ROI for controls in blue with the ADDT in the darker blue, and then stroke participants in green with the ADDT in the darker green. ADDT again is sentence processing. EPDT is the emotional prosody processing. We did not measure any differences between the groups in the amount of activation for either task. So it doesn't seem as though that the stroke participants in the right hemisphere have any more or less activation than in the typical brain for either of these tasks. We then looked at the relationship between the number of active voxels for both tasks within individual stroke participants. And so we have the ADDT or sentence processing number of voxels active on the y-axis and the emotional prosody voxels on the x-axis, and we did not measure a relationship in any direction between more activity in one leading to less activity in the other. This was also true for control participants in separate hemispheres, which isn't shown here. So these two tasks don't appear to be related in terms of amount of activation. So we then looked at what about when we make both tasks be equal in size, how overlapping are they spatially? And so we again have our top voxel analysis pipeline. And so here I'm just showing examples of the unthresholded activation maps for a few individual participants. That's why they look kind of crazy, because they're unthresholded. In blue is the sentence processing or ADDT activation, and in red is the emotional prosody or EPDT activation data. So for each control participant, we take their left hemisphere sentence processing activation and flip it into the right, because we want to be able to compare it with their emotional prosody activation in the right. And then for each stroke participant, everything's already in the right. So we're able to just compare their right hemisphere sentence processing activation with their right hemisphere emotional prosody activation. We're again looking in anatomical regions of interest. So we have the full frontal lobe and the full temporal lobe to see where things end up recruiting space. We again have our 4 top voxel levels. And so this time we have averages across both tasks for all participants. And so we end up with, for example, 2,000 voxels in the frontal lobe. So it'll be 2,000 voxels for the sentence processing task in the flipped left hemisphere for controls, and 2,000 voxels for the prosody task in their right hemisphere. So we're comparing maps of equal size, and then the same size for the stroke participants. So when we compare a control's left hemisphere activation flipped into the right to their own right hemisphere activation, that's in a sense task symmetry for the two tasks. For the stroke participants, it's task overlap because it's in the same hemisphere without any flipping. And so I really wasn't sure what we were going to find here. And it was really interesting to observe that actually with a pretty surprising amount of consistency, the stroke participants showed less overlap than the controls showed symmetry. And so I'm showing you here the frontal ROI on the left and the temporal on the right. The y-axis again is the Dice coefficient mean across levels. And so, yeah, the individual stroke participants as a group had less overlap. And so it doesn't appear to be the case that when language flips into the right hemisphere, it just overlaps with emotional prosody like we would expect when we literally did that with controls. And it's true even at individual top voxel levels. So this isn't even just because we averaged across them or anything. It's like a very consistent relationship that the task symmetry is greater in controls than the task overlap is in the stroke participants at each level. And I'll show you some visuals of this task overlap. I hope that the colors look okay. So at the top, I just have 3 individual control participants where you can see their flipped ADDT, so again, sentence processing in blue, and they're unflipped, so right hemisphere emotional prosody processing in red, and the areas in the magenta are where they overlap. And then below that, we have 3 example stroke participants, where in blue we have the ADDT, which is just in the right hemisphere, and then also the emotional prosody task, which again is also just in the right hemisphere. And you can see that not only is there less pink in terms of overlap between the tasks, each of these participants has a slightly different way of organizing the activation for each of these tasks such that it's not very overlapping. And so this was a really interesting finding. And this is an attempt at showing this at a group level. So in the pink, these are penetrance maps again, so this shows the consistency of overlap among participants. So the controls are on the left and the stroke participants are on the right. And so areas that are hotter pink or orange or yellow are greater consistency. And you can see that the areas that the groups have in common include parts of the IFG, pars triangularis, orbitalis, opercularis, and part of the superior temporal sulcus. So this would be areas where there was high task symmetry among controls, and there was also higher task overlap among stroke participants. And so there's some areas of overlap, certainly, but it's a lot less consistent in the stroke participant group, which relates to the findings that there was just less overlap in general in the stroke participants. The images below that show for controls on the left and stroke participants on the right, where the activations separated for each task. So in order to do that, for each participant, I took their top voxel map just for the most lenient threshold because it had the most voxels. And I said, where are their top voxels for the ADDT, but not the EPDT? And I just subtracted them for each participant. So this shows on these maps areas where there was consistently separation, where the ADDT was active and the EPDT was not. And so where the groups have in common areas that were more active for sentence processing, more consistently active for sentence processing, there's a small part of the IFG pars orbitalis, so the blue area in both of these brains in the pars orbitalis. And then for the ADDT greater than the emotional prosody task in just the stroke group, we see an area in the posterior middle temporal gyrus, dorsal, IFG, pars triangularis, and opercularis, and you can't really see it here, but part of the anterior insula. And then finally, areas where there seemed to be consistent recruitment for prosody, but not sentence processing in both groups. There was mid to posterior superior temporal gyrus, and then a small ventral portion of IFG pars triangularis. And so in conclusion, based on the 2 studies that we did in the perinatal stroke survivors, we found that successful organization of sentence processing in the right hemisphere after a left hemisphere perinatal stroke likely involves recruiting a left hemisphere-like distribution of perisylvian areas, which was the finding from the 2nd study. And then from this study, it may also involve segregating sentence processing regions and emotional prosody processing regions in order for both to develop successfully. And so in just 1 additional minute before I wrap up, I just want to talk about how these functional subregions might form. So this is speculative, but just a way that we might think about how these regions may form. So this is a term from the computational modeling literature, competitive specialization, which refers to how different systems may develop to be tailored to different dimensions of input information. And so in particular, there are models called mixture of expert models. A lot of this work has been done in the visual systems for dorsal and ventral streams and how you have differences in object recognition of what and where and how. And so in these models, there are similar but non-identical neural circuits or experts that are exposed to input data for complex tasks. Over the course of several trials, the likelihood that a given expert or network will be selected for a specific task is either increased or decreased. So you can think about this being during development. One expert becomes increasingly specialized for one type of task, while the other expert becomes specialized for a different type of task because it's competitive in nature. And so it's possible that a similar series of steps might give rise to specialized systems for sentence processing and emotional prosody, either in separate hemispheres, as we see in the typical brain, or in subregions in right perisylvian cortex if you only have one set of perisylvian regions to do both with. And so I just want to take a minute to thank the pediatric stroke research team, which is led by Lissa, and in particular my mentorship team for all of their effort towards my dissertation work. So Lissa Newport, Peter Turkeltaub, and Anna Greenwald, as well as our co-authors and collaborators who have put a ton of work into this project over time. And I also want to thank the Cognitive Recovery Lab, which is Peter's lab, and that's where I'm doing my postdoc now. The Center for Brain Plasticity and Recovery is run by Lissa. The Center for Functional Molecular Imaging is where we do all of our scanning. The IPN, which is my graduate program, and then my thesis committee gave me a ton of helpful feedback throughout time about these various projects. Max Riesenhuber, Ella Stream-Amit, Anna Greenwald, and then my external member is Brad Schlager. So that's it.
That's awesome. You want to ask folks first to chat to ask questions? All right, we have time for a few questions.
First of all, pretty cool. But I guess something I was confused about is the just the whole like top voxel pipeline, and I didn't quite follow that, and so is like all four of those threshold levels are being taken into account in the ultimate maps you ultimately end up with.
Yeah, thanks for asking about it. I never get it quite right. I either like go into way too much detail or like just skim it. Yes, so what we do is for each participant, we will take their unthresholded activation map, and then we'll say threshold at p less than 0.01, how many voxels are active? And then we take that voxel count for each participant. So just for the frontal cortex, just the frontal ROI, and we'll say just for that threshold, how many voxels? And then we take that average across everyone. And then we go down to p less than 0.005, and we do the same thing. And so that average across everyone for each of those thresholds becomes the top voxel cutoff. And so depending on the study, like for the first study, for example, where it was the healthy development left compared to right hemisphere, we determine it in the left hemisphere, and then we apply it to both the left and the right. So it'll be like 3,000 average across everyone in the frontal cortex for just the p less than 0.01 threshold. That's where the average came from. And then we apply that number to both the left and the right in order to get the same size maps. And so we do, that's how we get the 4 different cutoffs. It's based on the averages across everyone for each of those p thresholds. And then we calculate the dice overlap at each of those cutoffs. And then the bar graph data that I show is the average of those dice coefficients. So each person has 4 dice coefficients. That refers to increasingly smaller maps, like stricter in size. Okay, thanks for asking.
What question? I'm waiting.
Anna's question.
That is like amazing work, and it's really a lot of work. I'm really interested for thank you for these results. I had a question about the age of your groups. I'm sorry I missed the information, but are both of your groups adults?
I didn't show it, so good catch.
So my question is, both of the groups adults, and do you think you would find the same results depending on whether you're looking at children or adults?
Is this the right study? Yes. So good question. I didn't show it because I was hiding it or anything, but it's just a detail I didn't include. So for both studies, we looked at individuals who are older than 10 years old. And the reason for that is because that's when language lateralization appears to stabilize in the healthy brain. And so we really wanted to look at a stable language system organization and how it's organized once it's stable. And so our stroke participant group had 14 people in it for the second study, 13 for the third study. And the age range was 10 to 25. For the controls, there were 11 of them. And again, they're the healthy siblings of the stroke group. And they ranged in age from about 11 and a half to 29. So looking at children younger than that, it's possible that we may see something slightly different. Just because it's possible either in the controls or in the stroke participants that language is not in a stable organization in terms of lateralization quite yet.
There could be, for example, that in the children, the right hemisphere. For example, the activation controls right hemisphere, basically the right hemisphere activation of kids who have a stroke.
I think that's possible. So because we're including older children and the whole idea of their lateralization being stabilized is that there's less activation in the right. And so if we were to look very, very young, it's, the right hemisphere is more active as we've seen in prior studies. And so those areas of stronger activation may overlap better with the right hemispheres of the perinatal stroke kids. Yeah, I think that's definitely possible. I do think it's also possible that right hemisphere activation is in similar places to the weak activation that we see in the adults. And so it's quite homotopic. It's just stronger in the younger kids. And so it may be the case that even though it's stronger, it's still kind of slightly not in the right place to be as well aligned with the right hemisphere activation in the stroke participants. Thank you for your question. That was a good question.
To ask a question that looks like Marissa, were you planning to ask one?
Oh, no.
Okay. I think I'm having, of course, like, I want to echo, it's just really, really interesting, and it's gonna take us some time to fully understand all the results, and they're super cool. I think I need a little help with study 3, because your emotional processing task is essentially all of language plus emotion, and you're looking at that subtraction between language and the sort of added on emotional piece. So I guess I can't resolve that with, what I'm going to call blobs of specialization that you showed in your hypotheses, because they're expected to process language in that. What you're really looking at is the added emotional component to that. So what does that say about functional specialization, other than you're just taxing the system a bit more to do this emotion? I don't think I fully landed on what you were trying to say versus this idea of why do you think it's functional specialization? Or is it cost? I guess that's what I thought you were going to say, is that language is coming at the cost of emotional processing, and now they've been segregated, but that's not what you ended up saying at the end.
Are you referring to like some kind of like a crowding sort of effect?
Kind of thing.
So interestingly, I think that there is a caveat with our interpretations, but it may be in the opposite direction of what you were saying, which is that the nature of the subtraction in the emotional prosody decision task is subtracting out neutral speech. So it really is like almost extra isolating just the emotional processing that's going on. And then we're comparing that activation for like really narrowing in on the emotional processing to any regions that are responsive to forward speech over reverse speech. So we might actually be sort of pushing these regions in the extreme opposite directions and saying what is really active for speech processing according to the ADDT, and then what is really only active for emotional processing, subtracting out anything else that might be going on in the neutral speech conditions. And so I think our interpretations really are limited to areas that do one or the other. And that's not to say definitively that I know everything that's going on while they're doing this task. But we're actually, I think, getting rid of areas that may be doing some of both in both groups.
Would you expect them the same results for both groups if you're pushing? I understand what you're saying, though. You're sort of pushing the tasks further apart, but if that is true, then even the controls should show that non-overlap, because you're really testing two mechanisms.
But the controls are using two different hemispheres. And so the left hemisphere areas are, for the forward over reverse speech, they're just in these nicely mirror image regions that show a lot of overlap when we compare them.
That real estate can use, hence there's overlap.
Yeah, seemingly. So I didn't go in with any expectation for whether we should see some specialization or whether we should see just you can do both in the same regions under these special situations. But it just seems like consistently across the group, we just see a lot less overlap, at least when we analyze it this way. And I think there's a lot more we can do in terms of the types of tasks that we can use and the way that we probe where information is represented and how specifically in terms of language sentence processing versus prosody processing, but even just based on the general averaging of activation for these two tasks in the subtraction method, we do see pretty consistent sort of subregions.
I mean, that makes me feel like there's some sort of conscious or subconscious or cognitive resource allocation that might be happening in the stroke kids because they don't have damage to the right hemisphere. However, they're not necessarily engaging the entire cortex for the same two tasks. Whereas it looks like when you did the symmetry analysis, there's enough of a swath that's being used, and it's almost as if there's some sort of neural resource allocation that might be happening, which is completely
Yeah.
interesting.
I think at least three things that I would love to see as follow-ups to this work would be, thank you so much, the connectivity for each of these as networks. So are they actually connected to separate regions where you see a sentence processing network that's clearly discernible from an emotional prosody processing network, which you can see in the healthy brain in the opposite hemispheres. So do we actually see that it's really like separate systems? A second thing is what prosody activation and this sort of weak shadow of language activation looks like in the healthy brain. So to what extent are they segregated in the right hemisphere in the healthy brain? Are they leveraging something like existing partitions, or is this a major effect of plasticity? I think to some degree it is plasticity because they are recruiting a left hemisphere like network there, but maybe it's working its way around prosody regions in a way that's actually reflective of something we do anyway in the healthy brain. Especially if we have these like language active regions earlier in life, we still have prosody regions earlier in life in the right hemisphere as well. So how are they sharing space? Or maybe that is a reason why lateralization changes. And then the third thing, which I will go into a rabbit hole with, so I will hold back from that, is how the structure of the right hemisphere might be changing in the stroke participants. So in order to have subregions or something, is there actually just like more structural real estate? Are there increased, folding or surface area or something in order to fit both in the same amount of space?
That's also happening. Yeah. Okay. Other questions? Yes. Oh,
I have a more general question on your work. How does this work has changed your vision of the plasticity mechanisms in adult stroke? Because you worked on it before, doing that, but now adult stroke. So I'm curious how all this work has changed.
Yeah, thank you for asking that. I was very excited. Peter and I were both very excited for me to look at sort of the weak shadow areas in older adults because they're more closely matched to controls, that is, more closely matched to the adults who have strokes, to see if maybe when you do use the right hemisphere after a left hemisphere stroke in adulthood, that it's like leveraging these weak shadow regions, or it's not, or it perhaps the outcomes are not as good because you can't leverage the typical left hemisphere regions. And so that analysis is still kind of underway. We're like tweaking it a bit. It is not as clean of a picture as it was with the stroke kids, mostly because adult stroke language activation outcomes, it's all so much more variable. Whereas we had this picture with perinatal stroke where it's like you're performing language successfully and you're all doing it in these right hemisphere regions that appear to be so homotopic. And so as perhaps I should have expected, there's a lot more heterogeneity in the adult stroke situation, but I'm excited to at least try to describe a little bit better what the adult stroke picture looks like and what the plasticity limitations might be. There is some interesting work looking into increasing right hemisphere activation after left hemisphere stroke in adulthood to see if it's just the amount of activity. Bruce Crossan has done a lot of that work where you sort of do like movement therapy or like melody therapy and things like that, or using TMS as well to increase the activation. And it seems like that actually shows some beneficial effects sometimes. So I think, I don't know, there's a lot of potential to dig into it more.